So we talked about in chapter 6, like basically thermodynamics, right? Talked about this idea of work. And in biology, when we say work, it's the ability to cause change. So life is always changing, right? That is true. There's always these chemical reactions happening where we had your catabolic reactions where you broke things down and you used the energy from breaking things down to fuel anabolic reactions or to just fuel just doing work in general. If we look at our, this puffin here, this puffin is eating, I think that's a fish. I guess. And like the chemical bonds that hold that fish together, if the puffin can digest that, break down that food, and then use that energy in cellular respiration, break down the food to then get energy in the form of ATP. And that's what we'll be going over. And then, we talked about with the different laws of thermodynamics, and I showed you like an energy pyramid where like the sun is the ultimate source of all energy. The producers is the biggest level of the energy pyramid. And then like you have like the consumers would eat the producers. Arrow goes to the stomach. If I draw a bunny rabbit, there's my bunny rabbit. And here's a plant. There's a plant. The arrow goes like into the stomach of the bunny rabbit. These are the producers. You have like your first consumers, and then you can have like your second order consumers. And anyways, and the reason why each level of the pyramid got smaller is because every time you transfer energy from one form to another, you lose energy as some form of heat. Okay? Yeah, so anyways, and then if I jump to the next slide, what are we doing here? What happened? There we go. That second bullet point is talking about this cycle that in photosynthesis, the products of photosynthesis are the reactants of cellular respiration. So in photosynthesis, we use light energy to make, when they say organic molecules, think glucose. We make glucose plus oxygen, and then those products become the reactants that we put into the mitochondria to do cellular respiration. And then the products of cellular respiration, the carbon dioxide and water, those are then the reactants that would be used back in photosynthesis. This is like 7th grade, like biology. This was like the main thing when you learned cellular respiration, photosynthesis. That's really about all you learned, and then like where they occurred, chloroplast, photosynthesis, mitochondria, cellular respiration. Then 9th grade biology, you kind of got like, you dipped your toes into like the details of like cellular respiration and photosynthesis, and it was probably really hard to track with it because you didn't have chemistry yet. But anyways, we make ATP in the mitochondria. We use that to do cellular work, and then some of that energy again gets lost as heat. Now for us, we can like take some of that heat and use it to like warm up our bodies. It's not just like completely wasted energy. Yeah, and then we'll have to regenerate that ATP once we're done using it, and that's when we would get back into like eating again. And anyways, that's the basic cycle. Yeah, so as I mentioned, catabolic pathways will give us that energy. Catabolic, you're breaking things down. We break down our food like glucose and use that energy to make ATP. Now, what I'm going to show you is something called an oxidation, a redox reaction, where so catabolic is breaking down. How we yield energy from catabolic reactions is by oxidizing organic fuels. Now, oxidizing, you ever heard this acronym oil rig? I don't know if it's just a Texas thing. When I was in chemistry, like our teachers taught us oil rig. O, oxidation. I is L, loss. Oxidation is loss. And by loss, we mean electrons. So if you oxidize something, so we're going to oxidize glucose, C6H12O6, and what we're doing is we're removing electrons from glucose. And those removal of electrons, we're going to transfer them to some other molecules. The transferring of those electrons from glucose to other molecules will eventually give us some ATP. Other part of a redox reaction, reduction, R, is gain. Gain of electrons. So oil rig. And I'll show you kind of how the reaction works here in 2 slides. So yeah, catabolic pathways, we produce ATP. Remember, this is an exergonic process. Anytime you do catabolism, where you take something organized like glucose, and then we end up making CO2, H2O, and then some ATP and heat, we're taking something more organized and we're breaking it into more molecules. We increase the entropy. That is going to be an exergonic process. Remember, exergonic, the delta G is negative. We're releasing energy. Fermentation, this is whenever, I'll go over fermentation and anaerobic respiration towards the end of chapter 7, but for now, just kind of the basic definition of fermentation is it's like a process of how you're going to break down a sugar like glucose, but in the absence of oxygen. So that's how alcohol is made, right? The bacteria in that alcohol and like the grain, whatever it is, like the grapes, whatever it is that's doing the fermenting, those bacteria, they still need to make energy, but they'll take the sugars in the grapes, or the sugars in like even the wheat, or whatever the alcohol is, whether it's like gin, whiskey, like what you put in a different carbohydrate, like a different kind of like starting like grain, and that'll make the different kinds of alcohol. But anyways, the basic process is still fermentation. I'll show you that reaction later on in chapter 7. Aerobic respiration, that's what we're focusing on really in chapter 7. That's the bulk of chapter 7. Aerobic respiration. Aerobic means you're kind of active. Respiration, you're breathing in. So aerobic, active, you're requiring oxygen. So in aerobic respiration, we're using oxygen to break down, by organic fuel, we're talking about carbon-based fuels like glucose, C6H12O6. Anaerobic, that AN means like the absence of, so you're like in the absence of oxygen. So it would be like, how do we break down fuels without oxygen? So fermentation and anaerobic respiration are very similar processes, and I'll show you those reactions later in chapter 7. Okay, so getting into cellular respiration specifically, there is an aerobic part and an anaerobic part to cellular respiration. Ideally, your cells want to do the whole process, aerobic and anaerobic. When would your cells have to do the anaerobic part? Default is you do both aerobic and anaerobic, but there's like a shortcut of how you can get energy when it's anaerobic. When might that happen? When might your body have to like cut out the aerobic part of cellular respiration and only do anaerobic? You're exercising? You're exercising, right? Your body, you start running out of oxygen. Your cell, your muscles and stuff start running out of oxygen, so you only can do the anaerobic part of respiration. That's when, you ever heard of lactic acid? Yeah? You've all felt lactic acid, like some of that soreness from it. Anyways, cellular respiration with, like, if you're just doing the anaerobic part, you'll produce lactic acid. I'll show you that reaction a little bit later on. Okay, so when we talk about cellular respiration, we mainly focus on glucose. The starting ingredient is typically shown as glucose, but technically we could do cellular respiration with carbohydrates, fats, and proteins. With fats and also proteins. But the fats and proteins, they're going to go in at a different pathway. Cellular respiration, there's tons of different chemical reactions that are involved with it. Think of it like a highway. Glucose will start at the very beginning of the highway. The fats and proteins, it's like you can have like an on-ramp into the highway. They'll hop into cellular respiration at a different step in the process. At the end of the chapter, I'll show you kind of where fats and proteins would enter into cellular respiration. Okay. The main idea of what we're doing in cellular respiration is we are transferring electrons from glucose, from C6H12O6, to, you're basically taking these electrons and the removal of those electrons will ultimately fuel the synthesizing of ATP. Okay? So it's easy to kind of get lost in some of these details, but the main idea is we're removing electrons from glucose, and we're going to use those electrons to make ATP. All right, but first I want to explain to you what is a redox reaction. Now, if you look at like the objectives for the AP exam, you don't have to actually like understand the redox reactions. They're not. This isn't like a chem class and be like, what is the oxidation number of this atom? And like they're not going to show you that. But to actually understand cellular respiration, cellular respiration is a redox reaction. So I'm going to show you I'm going to get into the details of it. If I lose you a little bit, it's not a huge deal. But I just want to make sure you kind of get the full story since it's AP Bio. You need to understand electronegativity. We talked about electronegativity in the very first unit. Electronegativity is basically, you can think of it like how badly these elements, that's not a Y, elements want electrons. So if you have a low electronegativity, you don't want electrons that badly. Or you could say the affinity for electrons, how much you attract, these elements attract electrons is very low. Now, if you're a non-metal, you attract electrons, you want electrons very badly, so you have a high electronegativity. To understand why, let me go to your favorite Bohr models. So if I do the Bohr model for lithium, lithium has 1 valence electron. Now, when are atoms the most stable? Remember? Yeah, the full, we call it usually the octet. You get a full shell of electrons. So if you're lithium, there's 2 ways you can get a full shell of electrons. You could either gain 7 electrons and have an octet, or you could just lose 1. I think we can all agree it's easier to lose 1 electron than gain 7. That is why lithium, and it's going to be a similar story for all your metals, that's why metals want to lose electrons, because that's the most efficient way to get a full shell of electrons. Now let's compare that to, say, fluorine, the most electronegative atom on the periodic table. So in fluorine's Bohr model, it's going to have 7 valence electrons. So if you're fluorine, if fluorine can gain 1 more electron, then it would have a full outside shell of electrons. We call it the valence shell. So that's why the non-metals want to gain, because it's easier to gain 1 than lose 7 in this case. All right, now you could have a polar bond. So if you look at water, oxygen has an electronegativity of 3.5. Hydrogen has an electronegativity of, I don't know what that is, of 2.1. So that's why the electrons in this bond, in a bond you're sharing electrons, the electrons spend more of their time around the oxygen than the hydrogen. All right, so keep that in mind as we talk about the redox reactions. In every redox reaction, well, first the basic definition of a redox reaction. Redox reactions are kind of looking at the transfer of electrons from your reactants to your products. How do electrons get moved in a chemical reaction? Okay, and in a redox reaction, I said earlier, if something, oxidation would be whatever substance loses electrons. That's the oil in oil rig. Oxidation is loss. Reduction is whatever substance gains the electrons. We would say that thing is reduced. So reduction, reduction is gain. Let me show you an example. Here, if we should look at this ionic reaction, you have Na. Na is a metal. And chlorine is a non-metal. I just showed you from those Bohr models that metals want to lose electrons. So that's why sodium is going to want to lose an electron to be stable. Chlorine wants to gain electrons to be stable. That's why you get NaCl. But if we analyze this ionic compound that we made, NaCl, because sodium lost electrons, we would say it became oxidized in this reaction. And chlorine, because it gained electrons, we would say chlorine became reduced. Okay? Now, if you became oxidized, we call you the reducing agent. So it's backwards. If you got oxidized, you are the reducing agent. You're like doing the reducing. If you got reduced, you are the oxidizing agent. You are oxidizing something else. Agent. Okay. Again, on your test, on the AP exam, they're not going to like show you a chemical reaction and be like, which of the following is the oxidizing agent in this reaction? It's just to actually understand, like, I want you to understand what I mean when I say we oxidize glucose. That's more of the purpose behind this. Okay. Now, showing you the general formula. X and Y, those would just be elements. Xe would be like, you have the e to the minus, that's an electron. Xe, this would be saying this is whatever element got oxidized. This electron here is being transferred to Y. Y got reduced because it gained the electron from element X. And because X gave up the electron, it got oxidized. So there you go. All right, first two bullet points here. As I said, whatever got oxidized, whatever is donating the electrons, that's the reducing agent. Whatever accepted the electrons or got reduced, that is, I'll write that there. That whatever got reduced, that's the oxidizing agent. Whatever is the donor, that's what got oxidized. That is the reducing agent. That's backwards. Now, the reaction I showed you there, that was an ionic reaction where there was a complete transfer of electrons. Redox reactions can also happen in covalent bonds. Remember, a covalent bond is when it's a nonmetal plus a nonmetal? Okay? So you can have redox reactions in covalent bonds. This would be where, like, you have, maybe you have like a nonpolar bond, and then you make a polar bond. Right? It's still a covalent bond. Still, there's not a complete transferring of electrons, but you end up having a more polar bond that's created. I'll show you an example that'll make a little more sense. So here, this slide, this is a great slide. If you understand this slide, I am very happy. I would call it a successful day. So here we have, they use methane. Well, what we're looking at here is a combustion reaction. So cellular respiration is a combustion reaction. Combustion reaction is like when you you involve oxygen. So like when you start your, like, if you have a gas-powered car, you're doing a combustion reaction. When you light a candle, that's a combustion reaction. When you do cellular respiration and you add oxygen, you have your carbon-based fuel, your glucose, and you add it with oxygen, you are combusting is a combustion reaction. Okay? So here, CH4, I could change this to glucose, C6H12O6. But they're just showing us methane because to see how the electrons are moving, it's a little bit easier to understand. So here, methane, CH4, becomes oxidized. It's going to, and when I say becomes oxidized, we're focusing on the central atom. So carbon, and oxygen. Focus on carbon here. Carbon is going to become oxidized here because if you look at methane, carbon and hydrogen have about the same electronegativity. That's why the electrons are like in between. Okay? The electrons are being they're not being pulled more to the hydrogen nor toward the carbon because each of these have about the same electronegativity. But now, whenever this carbon becomes carbon dioxide, oxygen is the second most electronegative atom on the periodic table. So whenever we take the carbon from methane and pair up the carbon with oxygen and carbon dioxide, now those electrons are going to be super attracted to the oxygens. These are carbon to hydrogen, that's a nonpolar bond, nonpolar. Carbon to oxygen, that's a polar bond. So basically we took nonpolar bonds here and we made them polar. And what that does is that is technically a redox reaction because we are changing how the electrons are being shared. Then if you look at water, focus on oxygen here. The oxygen in our reactants, because oxygen in nature is O2, it's two oxygen molecules paired up, each oxygen molecule has the same electronegativity. The electrons are equally shared. But then once you pair up the oxygen with hydrogen, oxygen is way more electronegative than hydrogen, so now the electrons are going to be sucked into the oxygen. I hope that's making sense. Yeah. All right, now I'm going to go another level. This says energy is released. And like when I was in school, like teachers kind of just say this, and it was like, next slide, moving on. And it was like, well, how are we releasing energy? You know, it was like, and you might say, oh, well, it's an exergonic reaction, right? Delta G is negative. And you're like, all right, that's a little more clear, right? Like if I do the energy diagram, you know, I could show you an energy diagram and be like, all right, the reason why I release energy is because the products here, if we have potential energy on the y-axis, and like, you know, the energy diagram would look like this. This would be CH4. Here would be my CO2 plus my water. The reason why I release energy is because, you know, the products are up here at a higher potential energy. The reactants are at a lower, so the delta G is negative. That's why we release energy. That's not actually why you release energy. The reason why we release energy has to do with the electrons. So stick with me here. I think it's kind of a cool story. This molecule here, methane, the potential energy is higher. And that's what you see on the energy diagram. The reason why the potential energy is higher is because the electrons are not committed to one atom or another. So I'm going to say that again. The reason why potential energy is higher is because the electrons are not committed to one atom or another. All right? Now, going to the products, carbon dioxide and water, what do you notice about their electrons now? Now they are committed. Now these electrons are committed to the oxygen. They're committed to that oxygen. So that's why the potential energy goes down. Whenever the electrons, anytime the electrons go from a bond where they're being more equally shared to a more polar bond where they then get sucked into that more electronegative atom, you have a decrease in potential energy. That's the delta G, and energy gets released. Lower potential energy, this is more stable. Right? This is going to have higher entropy. The universe favors that. It is more stable because these electrons are attracted to the more electronegative atom. Okay? If I lost you there, that's okay. The AP exam's not going to go into that level of detail. I just wanted to show you, like, that's what's actually happening in these redox reactions. So when I say, you know, this got oxidized and that's why it's losing energy, this is why what's actually happening with the electrons. So I'll keep it moving. And that's what this bullet point is just saying here. In a redox reaction, we're going to move electrons anytime we move electrons from a less electronegative atom towards a more electronegative atom, we're always going to release chemical energy. Delta G is minus, and we can use that released energy to do some sort of work. In the case of cellular respiration, that work is to make ATP. Okay? All right. Now, during cellular respiration, the fuel is not going to be methane. It's still going to be a combustion reaction, but now the fuel is going to be glucose. This is going to be C6H12O6. But this is still a carbon-based molecule. Now, the reason why, like, you probably heard, or let me ask you this. Why is if I ate a fist full of like serving and this was like a donut versus say like maybe something that's like a protein like a chicken breast, why would the donut have more energy than say the chicken breast, more calories than say the chicken breast of the same size? Antigone?
It has more CH bonds.
Good. If you look at the structure of fats have more carbon to hydrogen bonds than do proteins. And you're like, okay. So what? Going back to this, carbon to hydrogen bonds, there's a lot of potential energy in these bonds because the electrons are not committed to one atom or the other. That's why you'll hear it said there's more energy per like gram of fat than per like gram of protein. Same story for your carbohydrates like glucose. There's more carbon to hydrogen bonds, so there's more non-polar bonds, being another way of saying it, that we could turn into polar bonds, thus releasing energy. All right. Anyways, and same for gasoline. That's why like gas, anything that's like a carbon-based fuel that has all its carbon to hydrogen bonds, a lot of energy is released from those bonds. Yeah, this is just saying like what we're doing in cellular respiration is we're going to take the electrons from glucose and we're going to use those electrons to create some ATP. They say transfer to oxygen. So I said oxygen was the second most electronegative atom on the periodic table. Oxygen is going to be like the vacuum that pulls all these electrons from glucose to then make ATP. It's at the end of what we call the electron transport chain. All right. So this slide, I wanted to show you kind of the same thing that I showed you with like the methane molecule. So I copied this from a couple slides ago, and showing you that we're doing the same thing now, but now we're changing this. Instead of it being CH4, now it's glucose. And this is the structure of glucose. What do you notice about the structure of glucose? That'd be interesting for a redox reaction. And it would kind of get into what Intignia just said. Will, think you know?
It has CH bonds.
CH bonds. There you go. Look at that. Carbon to hydrogen, carbon to hydrogen, carbon to hydrogen, carbon to all those C to H bonds where the electrons are not being committed to one atom or the other in the bond. That has a lot of potential energy. So here in glucose, high potential energy, right? It's higher up on our energy diagram. So whenever we oxidize and we strip away those electrons and stick them with a more electronegative atom like oxygen, that releases energy. All right. Hope that makes sense. And this diagram is basically what I drew up there, but then I added something else here. It begs the question, like, all right, you're saying that oxygen really wants to strip the electrons from your carbon-based fuel. The question is, well, why? And then you said that's an exergonic reaction. Well, why does that not just like happen like instantaneously? In other words, why has my car not exploded yet? Like, why? Like, I have a bunch of carbon-based fuel, like petroleum inside my car. Why is not just combined with all the oxygen and then just exploded? The reason is this: the activation energy. So the activation energy prevents us from releasing all that energy. Now, our body heat isn't high enough where we can like naturally make these reactions happen. So that's where we have enzymes. Our enzymes, like we talked about at the end of chapter 6, our enzymes lower that activation energy so that we can get that reaction going, so that we can ignite that glucose and get the energy out. That's the idea of the ignition on your car. Whenever you turn that key, the spark plug is providing that activation energy needed, and when you combine the oxygen with the fuel in your gas tank, you then create that spark, you meet the activation energy, then you can release all the energy and drive your car. Okay. I don't know why this slide is here, honestly. Same slide, I already went over this, but it's here. Moving on. Now we're entering the stuff where it's like, you need to know all of this, okay? As if I lost you with some of the redox stuff, it's not a huge deal. I just want you to know the full story. This is where it's like, all right, this is like what you need to like make sure you know all this stuff here. In cellular respiration, we're releasing a ton of energy here. A lot of energy is released, but if we release all of that energy at once, that's like an explosion. And like explosions might be kind of cool sometimes, but like that isn't very useful for making ATP because it happens and all of a sudden it's gone. So in cellular respiration, we do a series of steps so that we can like slowly utilize that energy. Controlled explosions, that way we are able to like harness all that energy from glucose and we don't just release it all at once and then we miss a lot of it. All right, so by organic compounds, remember they're thinking glucose, C6H12O6. We're going to take the electrons from glucose and we're going to transfer it to this molecule called NAD+. So we're stripping electrons from glucose. And you got a bunch of electrons going from glucose to NAD+. NAD+ is a coenzyme. It's something that coenzymes will work with enzymes to make them more efficient. We call NAD+ because it's taking electrons. It's an electron acceptor. And if you're an electron acceptor, if you're accepting electrons, you're getting reduced, you are an oxidizing agent. Another electron acceptor in cellular respiration is FADH. It's less common, but it will also do some electron accepting. So whenever we take these electrons from glucose, we'll form NADH. Now, hydrogen as an atom has how many protons and electrons? 1 proton, 1 electron. So whenever I say we're transferring electrons from glucose to NAD+, what that'll look like chemically typically is the addition of a hydrogen atom. That's why NAD+ becomes NADH. NADH is the reduced form of NAD+. Okay. All right, now, there are enzymes that will help facilitate the removal of electrons from glucose to the NAD+. Those electrons are called dehydrogenases. Now, the name actually makes sense. DE, that means the removal of. So removal, well, the removal of what? Of hydrogen. That's nice. Hydrogen, remember ACE is referring to an enzyme. So it's an enzyme that removes hydrogen and gives that hydrogen to NAD+, thus making NADH. Cool. And then there's also, in this process, we're also going to end up releasing another hydrogen ion. You'll see how this is used at the very end of cellular respiration, but this will then go through this proton pump that will then be used to make ATP. A lot of this, I know this is a lot right now. It'll start to connect together as we go through these in more details. So just kind of hang with me here. I want to talk a little bit more about your NAD molecule. So the full name of NAD is nicotinamide adenine dinucleotide. That's it. That's all it is. Okay. Now, the name is actually not as scary as you might think. Nicotinamide is a nitrogenous base. Where do we see nitrogenous bases? What biomolecule had nitrogenous bases? DNA, RNA, your nucleic acids. So here's nicotinamide. This actually isn't like a nitrogenous base that would be used in DNA or RNA, but we still call it a nitrogenous base because of all the nitrogens. Anyways, and it acts like a base. It's going to accept hydrogens. Anyways, what do you notice about this, and this? If I combine that, and that together, that should remind you of something from Unit 1. Remember? It's similar to ATP. Well, like thinking bigger how do we categorize that part that was part of Kai? Remember? A nucleic acid. This is a nucleic acid. This is you're like, I wonder what nucleic acid this is. Maybe it's adenine. Because it's nicotinamide adenine, and you're like, well, wait a minute, this kind of looks like 2 nucleotides that are, together. Di, that means 2. Nucleotide. Nicotinamide adenine dinucleotide. It actually does make sense, believe it or not. It wasn't just like arbitrarily named. So you won't have to like draw this structure, but understanding that story here. This is really just essentially two nucleotides together. So I want you to know that basic structure here. Now this is showing the reaction that I said in words on the previous slide, where we take glucose from our apple, we're going to strip away electrons. So we have all these electrons coming from our glucose. In biology, how we're going to represent those electrons is usually with like hydrogen. We're going to split apart that hydrogen into the electron and the proton that make up the hydrogen. All right, here's an electron, there's a proton. They both came from that hydrogen. We're then going to attach the electrons to our NAD here, and by doing that, how we attach the electrons to the NAD is by adding this hydrogen. So in other words, NAD plus becomes NADH. That's the end of the story here. That's really what I want you to get out of this. NAD plus is an electron shuttle. This NADH, we're taking the electrons from our glucose and we're going to shuttle them to the electron transport chain. And in the electron transport chain, we remove the electrons from NADH and then we'll make ATP. Okay? All right. I'll keep it moving. Hang with me. And that's what the first bullet point is saying. NADH will eventually pass those electrons down the electron transport chain to make ATP. In that electron transport chain, each molecule is going to have a higher and higher electronegativity. That will then why that's important is it'll function kind of like a vacuum, pulling the electrons from the NADH. Let me go to it. So here, this is what I'm saying. This molecule would have a high electronegativity. This would have a higher. That would have a higher, and this would have the highest electronegativity. Okay? That way we're pulling the electron, we're wanting to pull electrons down the chain, removing the electrons coming from the NADH that we made from the glucose. Okay? That's what they're trying to say in this bullet point here. And again, this isn't a control, we do this step by step, so it's not just one explosive reaction that we can't get all the energy from. And the purpose here is to make ATP from the ADP. Remember, you get the ATP from the ADP that you had used previously. Okay. This is just showing like if we did, the reaction just in one step, you'd release a ton of energy, but it wouldn't be very helpful. So then we do it in a controlled step-by-step pathway. Okay. The last slide I want to go over is just this. I know there's a couple other slides on there. I want to end with this slide. This is like kind of the freshman bio level understanding you kind of need to have of cellular respiration, where it was like, what are the inputs? What are the outputs? How much ATP do you make? You need to know that in AP bio, along with some more details we'll go into. But for now, this is the basic story we're going to get into after the break. The first part of cellular respiration is glycolysis. In glycolysis, we take our glucose and you take glucose. It is a 6-carbon molecule, and you make pyruvate. That is a 3-carbon molecule. You just like chop glucose into 2 3-carbon molecules. That's glycolysis. And when you do that, you will make a little bit of ATP. I'll explain substrate level phosphorylation after the break. I don't want to burden you with that today. You need to know where these processes happen. That's going to happen in the cytosol. And some of those electrons, when we do this reaction, we're going to produce some electrons that got attached to NAD+, making NADH. Okay? Then you take that pyruvate, and now you go into the mitochondria. In the mitochondria, and this is technically, these two processes here are in the matrix of the mitochondria. We take our pyruvate and we're going to oxidize our pyruvate. We're going to remove even more electrons from our pyruvate, our 2 3-carbon molecules. And when we do that, we will generate more NADH and more FADH2. And you make acetyl-CoA, which goes into what's called the citric acid cycle, sometimes called the Krebs cycle. aka the Krebs cycle. And we do more oxidation where we'll make more NADH, more FADH2, and you also will make a little bit of ATP, but just a little bit. Then we go to what's called oxidative phosphorylation, where now we're going to we're oxidizing the NADH and FADH2, removing the electrons that were stored. Think of these as storages of electrons. Remove those electrons and use them in the electron transport chain to make ATP. That's the basic story. So yeah, that's all I got. Let me stop the recording.